We all can see this, correct?
Yes.
Yeah.
Mm-hmm. Okay, here we go. Developmental biology, the study from fertilization of a mature egg by a sperm to the formation of an adult. There are 3 periods involved: the embryonic period, the fetal period, and the neonatal period. The embryonic period is from fertilization through the 8th week of development. The fetal period begins at week 9 and continues until birth. The neonatal period basically is the first month of a baby's life outside of the womb. I want you to know all of those periods. Okay, a man and a woman have sex, unprotected sex. They could be married, they could be single, they want a baby, they don't want a baby. It doesn't matter. For about 7 hours after ejaculation, sperm are not capable of fertilization. They go through a process called capacitation. Capacitation is when the sperm is involved in actions that undergo a series of functional changes that causes their tails to beat more vigorously and prepare them to not only enter but fuse with the plasma membrane of the egg. And remember, a secondary oocyte is known as the egg. Only capacitated sperm are capable of entering and fusing with the plasma membrane of the egg. What happens is once the one capacitated sperm penetrates the outside layer of the egg, this is known as the corona radiata. And then it gets into the zona pellucida, basically a clear glycoprotein layer between the membrane, which is the corona radiata, and the oocyte plasma membrane. It's all because of enzymes in the acrosome of the sperm and strong movements of the tail to help with penetration. Fusion of the sperm with the egg results in a word called syngamy, a process that blocks polyspermy. It blocks the fertilization of the egg by more than one sperm. And that's the word on this slide I want you to know. I want you to know what syngamy is, okay? I told you before, I'm not going to ask you any questions on meiosis and mitosis. That's not important. You should have learned that in AP1, and I'm not going to go through it again. Fertilization occurs when the haploid sperm and the haploid ovum merges to form a single diploid zygote. Remember, half of the genetic material comes from your father and half the genetic material comes from your mother. That's called haploid. The male is the sperm, the female is the ovum, and it forms a single diploid zygote. and the first embryonic stage begins. Where does fertilization take place? It takes place in the fallopian tubes, the uterine tubes. And it starts to grow, and it grows by mitosis. It divides, so it doubles every few hours. And there's a growing ball of cells that will eventually implant into the uterine cavity. It divides by mitosis. This is known as cleavage. It is a rapid mitotic division of the zygote, starting the first week of development. It forms blastomeres, progressively smaller cells that are produced by cleavage of the zygote. 3 to 4 days after fertilization, a solid ball of cells form called the morula. After that, about a day or two later, a blastocyst forms. This is what implants inside the uterus. Implantation is the attachment of the blastocyst to the endometrial part of the uterus, usually within 7 days after fertilization. Okay? Remember, I told you the lifespan of a sperm is 72 hours. So once the egg is fertilized, and it's fertilized in the uterine tubes, then it divides by mitosis and becomes a blastomere, then a morula, then a blastocyst, and the blastocyst is what implants into the endometrium of the uterus, usually within a week. Okay? And we just said this. This is just a review of what we said. Fertilization, cleavage, morula, blastocyst, and implantation. All right? The blastocyst attaches to the endometrium and burrows its way in. It is now a parasite. I know it's hard for you to believe that the fetus is a parasite, but it is a parasite. It lives off the nourishment produced by the hormones in the mother's blood. And the one hormone that prepares the uterus for implantation is progesterone. Without progesterone, there would be no implantation. So it's very important to understand how the blastocyst attaches to the endometrium of the uterus. The growth of the embryo, now it's called an embryo, okay? The growth of the embryo accelerates and passes through a number of stages in the formation of an embryonic disc and 4 extra embryonic membranes. The embryonic disc is the fetus. The extra embryonic membranes are called the amnion, the yolk sac, the allantois, and the chorion. We're now talking about the fetus. The trilaminar embryonic disc is the fetus. It develops into 3 primary germ layers. All of these tissues is what the fetus is derived from. The ectoderm is the superficial area. The mesoderm is the middle, and the endoderm is the inner layer. The ectoderm differentiates into the brain, the nerves, and the skin. The mesoderm basically is all the connective tissue, the blood, and all the muscles and bones. The endoderm basically is your organs, the digestive tract, the respiratory tract, all the other organs, and the endothelial lining of blood vessels. I would like for you to know all of these layers. The extra embryonic membranes, there are 4 of them. The amnion, the innermost, is what forms the amniotic cavity. It forms the bag of waters that the embryo is surrounded by. The yolk sac becomes the site of early blood formation. The allantois forms the umbilical cord, the attachment to the mother. The chorion, the outermost extra embryonic membrane, forms the fetal part of the placenta and produces a hormone called HCG, human chorionic gonadotropin. Why is this important? Because this is the pregnancy test. This is what women do when they go to the pharmacy and buy a pregnancy test. They test their urine for human chorionic gonadotropin. If you see a positive, that means you're pregnant. If you see a negative, that means you're not pregnant. It's testing human chorionic gonadotropin that is produced by the chorion. Here are the layers. Okay, here are the layers. Here they are. I would know all the 4 extra embryonic membranes, and I certainly would know the layers of the embryonic disc. The placenta. The placenta separates the mother from the fetus. The placenta is the site of nutrient and waste management. But it does prevent mixing of fetal blood with mother's blood unless there is a leak. And we talked about that leak when we talked about the fetal syndrome where you have hemolysis during pregnancy in the bloodstream, in the blood system lectures, okay? The placenta also produces hormones, and the 3 hormones it produces is human chorionic gonadotropin, estrogen, and progesterone, okay? So the chorion and the placenta both produce the pregnancy hormone, human chorionic gonadotropin. The placenta is formed by the chorionic villi of the embryo and the decidua basalis of the endometrium of the mother. Throughout the remainder of pregnancy, the placenta is responsible for maintaining the uterine environment for the fetus, and it prepares the mammary glands for breastfeeding. That is the embryonic period. Now let's talk about the fetal period. The fetal period begins at the 9th week and goes through the 40th week. This is when all the tissues develop. All the tissues develop. Fetal growth is remarkable. It makes you really believe in God. Okay? Everything grows. You don't have to know when things grow. That's not important. It's only important if you take a course in embryology, human embryology. Okay? But by week 34, the fetus is ready to be delivered. Now, it won't be delivered until the 39th or 40th week, but it prepares itself for labor and delivery. These are the developmental stages. You don't have to know the developmental stages. You have to know the periods, the periods. So the first trimester, which is the first 3 months, the corpus luteum secretes estrogen and progesterone, necessary to maintain the lining of the uterus and prepare the mammary glands to secrete milk. Once the chorion develops, it begins to secrete human chorionic gonadotropin to maintain not only the corpus luteum, but to maintain the uterus. From the 3rd month on, the placenta job is to maintain the uterus lining because it produces 3 hormones: estrogen, progesterone, and human chorionic gonadotropin. You don't have to know about human placental lactogen. That is not important. Neither is CRH. Those are not important hormones. But relaxin is one. Remember, relaxin originally comes from the ovaries. It's part of RIPE, relaxin, inhibin, progesterone, and estrogen. But once you're pregnant, you don't need your ovaries anymore to produce hormones. So relaxin is produced by the placenta. It increases the flexibility of the pubic bone and helps dilate the cervix during labor. And that's the reason you have relaxin. Here it is, relaxin. It allows you to deliver that baby. Okay? That is important. That is important. All right? So what are the changes during pregnancy? Well, if you've ever been pregnant, you know the changes, but let's go over them. Obviously, you gain weight because you got a parasite in your body. Okay? You store more protein. You store more triglycerides. You store more minerals. Your breast enlarges. You get spinal lordosis because you now have back pain because your belly gets bigger because you're carrying a lot of extra weight. A woman who is pregnant increases her blood volume, increases her red cell mass because there are more demands put on her from the parasite, the fetus. So a female that's pregnant, cardiac output rises by 20 to 30%. Your minute ventilation increases up to 40% to supply more oxygen to the fetus. Your digestive system and your brain appetite centers are liable for increased energy loads imposed by the fetus. You urinate more because there's pressure on your urinary bladder by your enlarging uterus. So you get increased frequency, increased urgency, and a thing called stress continence, that when you're in stress, you may wet yourself when you're pregnant. What is labor? Labor is the bringing of the fetus to birth, otherwise known as delivery. The onset is determined by several hormones, not only the placental hormones, but also the fetal hormones. The levels of estrogen rise very quickly at the end of pregnancy. To overcome the inhibiting effects of progesterone on uterine contractions, progesterone stops uterine contractions. Estrogen rises to overcome that inhibition. High levels of estrogen also increase the number of oxytocin receptors in the myometrium. That's where the uterine muscle fibers are. This makes the uterus more sensitive to oxytocin. Remember, oxytocin is the ejection hormone. So oxytocin, which is produced in the hypothalamus, stored in the posterior pituitary, and released when you are in labor. So when you're in labor, there are high levels of oxytocin in your blood, and at the same time, relaxin from the placenta is released to dilate your cervix and relax your pubic bone. And then you go into labor. And labor is a positive feedback mechanism. The release of oxytocin is all due to positive feedback, and it stimulates the uterus to contract more. Labor is divided into 3 stages. The first stage, and the longest phase, is the stage of dilation, typically lasting 6 to 12 hours, but can last for 24 hours. If you are in labor and you're dilating and you're not dilating fast enough, sometimes the obstetrician will rupture your membranes. They will rupture your amniotic sac so that you can quicken that stage of labor. So the stage of dilation is the longest stage. Stage 2 is expulsion. The fetus now is in the air, outside of the woman's body. Okay? Contractions force the fetal head into the cervix. Stretch receptors release more oxytocin, positive feedback, more contractions, more stretching. And when the baby is delivered, the cycle stops. The third stage is the placental stage. This is known as afterbirth because it's after the baby is delivered. The placental stage is delivery of the placenta and is expelled by the uterus. So those are the three stages of labor: dilation, expulsion, and placental stages. All right, now the baby's out. Lactation is the production and ejection of milk from the mammary glands. Now, I'm going to take issue with this definition. Lactation, in my mind, is only production of milk, not ejection of milk. So I don't agree with this definition. And my definition is lactation is the production of milk. The principal hormone in milk production is prolactin. And if you remember, prolactin comes from the anterior pituitary gland. Prolactin levels increase as pregnancy progresses, but there is no milk production because since the baby is still inside the mother, progesterone inhibits the effects of prolactin. After delivery, the inhibition of prolactin by progesterone is removed. Okay? The milk ejection reflex. The milk ejection reflex is initiated from touch receptors in the nipple and stimulated, sending sensory nerves to the hypothalamus, causing the release of oxytocin from the posterior pituitary. Remember, oxytocin is produced in the hypothalamus, stored in the posterior pituitary, and is the ejection hormone. It not only ejects the baby, but it also ejects milk. And it also has a third function. It is the love hormone. It forms a bonding, not just with the baby and mother, but also with the husband and wife or the sexual partners. Okay? So that's the milk ejection reflex. Before the appearance of true milk on the fourth day, the lactating breast secretes for the first 3 days a cloudy fluid called colostrum, which contains important antibodies. Important antibodies. Remember, especially IgA. Remember, IgA is one of the antibodies that is in breast milk. Okay? So it does give you antibodies. It gives you other antibodies too. All right? So that is important. That's important. Before I go on, let me ask a question. Let's say you work as a doctor or a nurse or a medical professional, and a man comes into your office complaining that there is milk coming from his breast. What do you think the problem would be if a man was developing milk coming from his breast?
Steroids.
Who said that? Somebody said something.
Patricia, I said steroids. I would think steroids.
He said he was taking steroids?
Possibly.
I would say no.
Okay.
What controls milk production? What hormone?
Prolactin?
Thank you. Where does prolactin come from?
The pituitary gland?
The anterior pituitary gland. Be specific.
Okay.
The anterior pituitary gland, correct?
Yes.
So if I have a tumor of my anterior pituitary gland, and the tumor is involving my lactotrophic cells, I will produce more prolactin, and I will have milk coming out of my breast. Okay? So, and that has occurred. I've seen that once or twice in my 40 years of being an ER doctor. Scary. And these guys had tumors of their pituitary gland. All right? All right. The baby is born. The baby immediately takes a deep breath for the first time ever. Inflates their lungs, cries. Now the baby is breathing air for the first time ever. So those are the respiratory adjustments of the neonate. Now you are in the neonatal period. The first 30 days of the baby's life is called the neonatal period. They breathe for the first time. They also have cardiac changes. Remember, in the cardiac system, the cardiovascular system, you learned that when the baby is inside the mother, it doesn't need to breathe. So there is a foramen ovale. By the way, this is spelled wrong. A foramen ovale that is between the right atrium and the left atrium, because the blood does not need to go to the lungs because the baby's not breathing. So there's a hole there in the fetus. When the fetus starts to breathe, that hole closes. It takes about 30 days for that hole to close. Okay? Because the baby now is breathing, doesn't need the shunt anymore away from the lungs. All right? So the foramen ovale closes. All right? All right. Any questions on the fetus, the periods, labor, lactation, ejection? All right. If not, we will talk about genes and inheritance, and then we'll call it a day. This is an overview of genetics. If you want to know more about genetics, I would recommend you take a class in genetics. It's fascinating, absolutely fascinating. The branch of biology that deals with inheritance is known as genetics. The diploid number of chromosomes is in the fertilized ovum. One chromosomal pair comes from the mother and the other comes from the father. These are called homologous chromosomes. Each pair in the genetic sampling of a human cell is a homologous pair. So all of these 1 through 23 pairs are all homologous. This could, I'm not sure what these are, but this could be skin, this could be eye color. It's all homologous. One from the mother and one from the father. They are the same trait, homologous pairs. Each of the two homologs contain genes that control the same traits, eye color, hair color, etc. Okay? Alternate forms of a gene that code for the same trait that are on the same location on homologous chromosomes are called alleles. And here is what we're talking about. We're talking about this allele on this gene that codes for this type of DNA. Now, this type of DNA might be blue eye color. Okay? So that's what we're talking about. We're talking about genetic information that appears on chromosomes. For example, and they use the greatest example, you already know about blood typing. So blood typing is homologous chromosomes. Each contain a gene that codes for a blood type with all of the possible alleles, A, B, AB, or O. These are all proteins. The possible results are all the children. Here you can see them. I think we went through this when we talked about blood typing. Okay? So you can see all the blood types of what each child would be. Remember, A and A, it's basically either AA or AO. So these kids from these two parents can only be A or O, etc., etc., etc. All right? A person with the same allele on homologous chromosomes, for example, capital A, or little letter A, is said to be homozygous for that gene. So this is, when you see big letters, capital letters, it's a dominant trait. When you see little letters, it is a recessive trait. And if it's AA or little a, it's known as homozygous. If there are different alleles on homologous chromosomes, dominant and recessive, it is said to be heterozygous. And it's for the same trait, Okay? So this is homozygous, and this is heterozygous. The genotype is what we've been talking about. It is the genetic makeup of the individual. A phenotype is what we see. We see the blue eyes. We see the brown hair. It is the expression of the gene. I wish it was that simple. It is not that simple. There are many factors that interfere with genotypic makeup of an individual and the eventual phenotypic expression of that gene. Okay? There are many factors involved. This is very simplified. An allele that dominates or masks the presence of another allele is the dominant allele. And usually the domination is capitalized. Here it is, big P. An allele whose presence is completely masked, little p, is the recessive allele. All right, so capital letters is dominant, small letters is recessive. A heterozygote, PP, big letter, little letter, has a different genotype from a homozygote, but both this and this give you the same phenotype. If dominant P was brown eyes and little p was blue eyes, the dominant trait will show up. So this person will have brown eyes and this person will have brown eyes. Even though they are different, one is a heterozygote and one is a homozygote, they have the same phenotype. Color eyes are still brown. So you need to know a Punnett square. We talked about Punnett squares when we talked about blood types. So you need to know what a Punnett square is, and this is a Punnett square. This is a Punnett square, okay? This happens to be skin color. This happens to be skin color, okay? So if say the capital letter A is brown and the small letter A is white, if you have a person who is A or this is dominant. This is all dominant. This is A and this is A. It's dominant, and you give a person whose egg is A and little letter A, you have dominant, which would be brown skin, homozygote, heterozygote, dominant, brown skin, heterozygote, dominant, brown skin, recessive, AA, white skin, and a white skin person is called an albino or albinism, okay? So that's what we're talking about. All of these three have the same phenotype, even though one is a homozygote and two are a heterozygote. The recessive gene combination will give you white skin. Aneuploidy, a word in which the individual is either born with an additional chromosome or a missing chromosome. And the classic additional chromosome is known as trisomy, and it's known as trisomy 21. Trisomy 21 will give you Down syndrome. Okay, Down syndrome, a common trisomy, a common trisomy. Instead of having 2 chromosomes, you have 3 chromosomes, 2 X's and 1 Y. All right, you have more. There they are. Like I said before, it's not that simple. There are many factors involved. Okay? If your mother and father smoked, you got trouble in your genes. If your mother and father are alcoholics or drug addicts, you got troubles in your genes. Okay? So there are many environmental factors that affect everything. Eye color, skin color, height, okay? If you're lacking minerals, if you're lacking vitamins, you have genetic problems. You have genetic problems. So many things affect genes, many things, all right? So let's say you're pregnant. You have a family history of a horrible disease. Let's say you have a family history of ALS, amyotrophic lateral sclerosis, otherwise known as Lou Gehrig's disease. And you want to know if your baby, while in the womb, is got those genes. And there is a genetic test for those genes. So a doctor will do a procedure called an amniocentesis, in which they stick a needle into your amniotic sac with ultrasound guidance, and some of the fetal cells within the fluid will be sampled. This is usually done between the 14th and 16th week of pregnancy to see if there is a genetic problem with your baby. It is recommended for all women who are pregnant after the age of 40, because after the age of 40, when you are pregnant, you are susceptible to many genetic diseases that you're not susceptible to when you're 18 and 20. Okay? So we can test for all kinds of diseases, genetic diseases, from an amniocentesis that is done between the 14th and 16th week of pregnancy. All right.